Hello there, and welcome to Biology for Sleep, a quiet place where science slows down, where curiosity is allowed to soften, and where learning is something you can do with your eyes gently closing. This channel is built for evenings like this, when the day has already asked enough of you, when the world feels a little loud, and when it might be comforting to let a calm voice carry you through something interesting, without demanding anything in return. You do not need to remember every detail. You do not need to stay awake. You only need to be here for as long as it feels comfortable. Tonight, we're going to explore something that quietly shapes almost every emotional experience you have. It influences how hopeful you feel in the morning, how steady your thoughts are in the afternoon, how heavy or light your mood feels in the evening, and how easily your mind lets go when it's time to rest. It does all of this without ever announcing itself. You never feel it directly, you never see it at work, and yet it is deeply involved in whether a day feels manageable or overwhelming. That something is serotonin. You may have heard the word before. It shows up in conversations about happiness, depression, anxiety, medication, and mental health. It is often described in simple terms as a feel-good chemical, but that phrase hides more than it explains. Serotonin is not a happiness switch. It does not pour joy into your mind on demand. It is not a mood button that gets pressed when life goes well. It is a messenger, a regulator, a coordinator, a small molecule that carries information between cells, helping your brain and body decide how to respond to the world. It plays a role in mood, yes, but also in sleep, appetite, digestion, memory, impulse control, and how you react to stress. It helps determine whether your thoughts feel steady or scattered, whether emotions rise slowly or all at once, whether your internal world feels balanced or unpredictable. It is involved in the background processes that make your mental life feel coherent. Most of the time, you're not aware of it. You wake up feeling a certain way. You respond to situations. You have reactions, thoughts, sensations, and all of it feels like you. But underneath those experiences, a vast network of cells is exchanging chemical messages. Serotonin is one of the most important of those messages. It does not dictate who you are, but it shapes how your inner landscape behaves from moment to moment. Tonight's journey is about understanding what serotonin actually does, where it comes from, how it works, and why it matters. Not in the language of textbooks or medical charts, but in a way that connects it to everyday experience. The goal is not to overwhelm you with terms or data. The goal is to help you see how something microscopic can have such a large influence on how a human mind feels. Before we begin, take a moment to notice where you are. What room are you in? Is the light dim or already off? Are you lying down or sitting somewhere comfortable? And what time is it in your part of the world right now? Evening arrives at different moments for different people, but the slowing down feels familiar everywhere. If this is your first time here, you might already sense that nothing here is in a hurry. If you find this style of exploration soothing, or if you enjoy learning in a way that doesn't demand sharp focus, you're warmly invited to like the video or subscribe. It helps this quiet corner reach other people who might need something gentle at the end of their day. There is no urgency in that, just a small invitation. Now, let's begin easing into the subject itself. Serotonin is a chemical messenger known as a neurotransmitter. That word simply means it carries signals between nerve cells. Your brain is made of billions of neurons, and those neurons do not touch each other directly. They communicate across tiny gaps using chemicals. Every thought, every memory, every sensation relies on this system. Electrical signals travel down a neuron, trigger the release of a neurotransmitter, and that chemical crosses the gap to influence the next cell. Serotonin is one of the most widespread and influential of these messengers. It is produced in several places, but most people are surprised to learn that the majority of serotonin in your body is not in your brain at all. Roughly 90% is made in your digestive system. It helps regulate how your gut moves, how it responds to food, and how it communicates with the nervous system. Only a smaller portion is made in the brain, but that portion has a profound effect on mood and cognition. The serotonin in your brain does not float freely in a pool. It is released in precise locations in tiny amounts by specialized neurons. These neurons originate primarily in an area deep in the brainstem called the Rafe nuclei. From there, their fibers spread widely, reaching regions involved in emotion, decision-making, memory, sleep, and perception. This wide distribution is one reason serotonin affects so many aspects of experience. Rather than creating specific emotions, serotonin helps regulate the tone and stability of mental activity. You can think of it as influencing how reactive or settled your internal state feels. When serotonin signaling is balanced, thoughts tend to feel more flexible, emotions less overwhelming, and stress responses easier to regulate. When it is disrupted, the mind can feel noisier, more rigid, or more easily tipped into distress. This does not mean that low serotonin causes sadness in a simple, direct way. Human emotion is far more complex than a single chemical, but serotonin participates in the systems that decide how strongly the brain responds to challenges, how quickly it recovers, and how predictable emotional patterns feel over time. In that sense, serotonin is less like a happiness generator and more like a stabilizer. It influences the background conditions in which emotions arise. It helps determine whether emotional waves feel manageable or chaotic, whether your internal environment feels calm enough to think clearly, and whether stress fades or lingers. Understanding this changes how we think about mood. Feelings are not just reactions to events. They are shaped by the biological context in which those events are processed. Two people can experience the same situation and feel very differently, not only because of personality or history, but because their internal chemistry responds differently. Serotonin is one part of that difference. As we move forward, we'll explore how serotonin is made, how it travels, how it interacts with other systems in the brain, and how it becomes linked to experiences like anxiety, depression, motivation, and sleep. We'll look at why light matters, why movement matters, why rest matters, and how modern medicine tries to influence this system. Each part connects back to the same quiet question. How does a brain decide what it feels like to be you today? There is no need to hold all of this in your mind at once. Let the information arrive at its own pace. Think of this not as a lesson to master, but as a slow unfolding of how something small becomes something meaningful. The details will build naturally, and nothing here depends on remembering what came before. For now, simply settle into the idea that your emotions are not floating in empty space. They are carried, shaped, and guided by physical processes happening inside you every second. And among those processes, serotonin is one of the most influential. We'll begin next by looking more closely at what this molecule actually is, how it was discovered, and why scientists became so interested in it in the first place. The story of serotonin starts not with happiness, but with blood vessels, digestion, and a surprising set of early experiments. The path it takes into the brain is a reminder that the boundaries between body and mind are far less firm than they seem. Serotonin did not enter science as a mood molecule. It was not discovered by psychologists trying to understand happiness or psychiatrists searching for relief from sadness. It first appeared in laboratories as a chemical curiosity linked to blood vessels and digestion. Long before it became associated with emotion, it was simply a substance that made tissues contract. In the late 19th and early 20th centuries, physiologists were deeply interested in how the body regulated itself. They noticed that certain tissues would tighten or relax when exposed to extracts taken from blood or organs. These effects were not random. Something in those extracts was sending instructions. Researchers began isolating and naming these substances, building the early foundation of chemical signaling in the body. One of those substances caused blood vessels to narrow. It made smooth muscle contract. It appeared in serum after blood clotted. In the 1940s, a scientist named Maurice Rapport helped identify and purify it. They named it serotonin, combining serum and tonic, reflecting its effect on tone in blood vessels. At that time, no one was thinking about feelings. Serotonin was part of cardiovascular research. It was a peripheral chemical, something that belonged to the body more than the mind. But as techniques improved, scientists began finding serotonin in unexpected places. They found it in the digestive tract, they found it in platelets, and eventually they found it in the brain. That discovery changed everything. Finding serotonin in brain tissue raised an immediate question. If this molecule existed in nerve-rich regions, what was it doing there? Neurons communicate using chemicals. If serotonin was present, it might be part of that communication system. Experiments soon confirmed this. Serotonin was not just floating passively, it was being released by specific neurons, binding to receptors, altering how other neurons behaved. This placed serotonin among a growing family of neurotransmitters, but it stood out because of how widely its influence spread. Some neurotransmitters act in narrow circuits, controlling precise movements or sensory signals. Serotonin's pathways, by contrast, were broad and diffuse. A relatively small group of serotonin-producing neurons sent branches throughout the brain. These neurons originate primarily in the brainstem, in clusters known as the raphe nucleus. From this deep, ancient part of the brain, fibers extend upward and outward. They reach into the limbic system, which is involved in emotion and memory. They reach into the prefrontal cortex, which shapes planning, reasoning, and impulse control. They influence the hypothalamus, which regulates sleep, appetite, and hormonal balance. They touch regions that process sensory input and those that coordinate movement. This anatomy alone suggested that serotonin would not be responsible for a single feeling or behavior. It would modulate many systems at once. It would not deliver specific content like joy or fear. Instead, it would adjust how those systems responded, how easily they activated, how long they stayed active, and how strongly they influenced the rest of the brain. Think of it as a regulator of tone rather than a generator of events. Serotonin does not decide what you think about. It influences how your thoughts unfold. It does not choose your emotional reactions. It shapes their intensity, duration, and stability. This distinction matters because popular culture often simplifies neurotransmitters into emotional buttons. Dopamine becomes the reward chemical. Oxytocin becomes the love hormone. Serotonin becomes the happiness chemical. These labels are catchy, but they are misleading. They imply that each molecule creates a specific feeling in isolation. In reality, emotions arise from networks of activity involving many systems at once. Serotonin participates in those networks by setting background conditions. It alters how responsive neurons are. It influences how signals propagate. It helps determine whether a stimulus triggers a strong cascade of reactions or a muted response. It can make the brain more flexible or more rigid in how it processes information. One way scientists came to understand this was through pharmacology. When drugs altered serotonin signaling, they did not create a single uniform effect. Instead, they changed patterns. Some people felt calmer, others felt more alert. Some noticed shifts in sleep or appetite before any change in mood. These broad effects reflected serotonin's widespread reach. To understand how this works, it helps to look at receptors. Serotonin does not act in just one way. It binds to many different receptor types, each with its own structure and effect. There are more than a dozen known serotonin receptors, grouped into families. Some excite neurons, others inhibit them. Some act quickly, others trigger slower changes inside the cell. This means that the same molecule can produce very different outcomes depending on where it binds. In one brain region, serotonin might quiet activity. In another, it might enhance it. In one circuit, it might promote patience and restraint. In another, it might facilitate learning or sensory processing. Because of this complexity, serotonin cannot be mapped onto a single emotional axis. It does not simply move you toward happiness or away from sadness. It tunes the system in which those experiences arise. Early animal studies revealed that altering serotonin levels changed behavior in subtle but consistent ways. Animals with disrupted serotonin signaling became more impulsive. They had difficulty waiting. They reacted more strongly to stress. They showed altered patterns of sleep and feeding. These changes resembled aspects of human emotional disorders, not in content, but in regulation. In humans, correlations soon emerged between serotonin function and conditions like depression, anxiety, and obsessive thought patterns. This did not mean that these conditions were caused by low serotonin in a simple sense. It meant that serotonin was part of the regulatory machinery that was not working as smoothly. One of the most influential developments came with the introduction of selective serotonin reuptake inhibitors, or SSRIs. These medications increase the availability of serotonin in synapses by slowing its reabsorption into neurons. They do not add serotonin to the brain. They alter how long it remains active after being released. The fact that these drugs could relieve symptoms for some people reinforced the idea that serotonin played a central role in mood regulation. But the time course was revealing. The chemical change happens quickly. The clinical effects take weeks. This showed that serotonin's influence is indirect. It does not simply flip a switch. It initiates gradual changes in how circuits adapt, how receptors respond, and how networks reorganize. This fits with what we now know about the brain. It is not static, it is plastic. Patterns of activity shape connections. Chemical signals influence gene expression, receptor density, and synaptic strength. Serotonin participates in these long-term adjustments. It helps shape the brain's response patterns over time. That is why serotonin is involved in both moment-to-moment regulation and longer-term emotional style. It influences how a single stressor feels. It also contributes to whether stress tends to accumulate or dissipate across days and weeks. Another layer of complexity comes from the body. As mentioned earlier, most serotonin is produced in the gut. Specialized cells in the intestinal lining synthesize it from tryptophan, an amino acid obtained from food. This serotonin does not cross into the brain because the blood-brain barrier prevents it. But it still plays a major role in bodily regulation. Gut serotonin controls how the digestive tract moves. It helps coordinate the rhythmic contractions that move food. It influences how the gut responds to irritation. It communicates with immune cells and with the vagus nerve, a major pathway connecting the body to the brain. This gut-brain connection has become an active area of research. Signals from the digestive system can influence mood, stress response, and cognition. While the serotonin in the gut is not the same pool as the serotonin in the brain, the two systems interact through neural and hormonal pathways. This helps explain why digestion, stress, and emotion are so tightly linked in experience. You have likely felt this connection. Anxiety can tighten the stomach. Anticipation can alter appetite. Illness can change mood. These are not metaphors. They reflect bidirectional communication between systems that evolve together. Serotonin sits at the center of this integration. It is not confined to one organ or one function. It is part of a distributed network that coordinates internal state. When people speak about chemical balance, they often imagine a simple scale with too much on one side and too little on the other. The reality is more like a complex control panel with many dials. Serotonin is one of those dials. Turning it slightly affects many channels at once, but the outcome depends on the existing settings of all the others. This is why two people can respond differently to the same medication. Their receptor distributions differ, their neural circuits differ, their histories of stress, learning, and sleep differ. The same change in serotonin availability can ripple through two brains in different ways. It is also why mood cannot be reduced to chemistry alone. Life events, relationships, habits, sleep patterns, and physical health all shape how these systems operate. Serotonin does not exist in isolation. It interacts with dopamine, norepinephrine, cortisol, and countless other signals. It responds to light exposure, physical activity, and nutritional state. It is embedded in a living organism. What makes serotonin particularly interesting is that it operates at the boundary between stability and change. It supports consistency in mood and behavior, but it also enables adaptation. It helps the brain learn from experience without becoming trapped by it. It moderates how strongly events imprint on emotional circuits. In this way, serotonin contributes to resilience, not in the motivational sense, but in the physiological sense. It influences how easily the nervous system returns to baseline after activation. It shapes whether stress lingers or resolves. It affects how flexible thinking feels under pressure. These are not dramatic effects. They are subtle. You do not feel serotonin rising or falling. You feel the result as a difference in tone, a day that feels manageable rather than overwhelming, a thought that passes instead of looping, a pause before reaction, a steadier sense of continuity across hours. This is why understanding serotonin is not about chasing happiness. It is about understanding the background conditions that make experience feel coherent. As we continue, we'll look more closely at how serotonin is made inside the brain, how neurons regulate its release, and how its signaling patterns change across the day. We'll see how light, sleep, and rhythm interact with this system, and why mornings and evenings feel different at a chemical level. For now, let this first meeting settle. Serotonin is not a magic ingredient. It is not a mood in a bottle. It is a quiet coordinator, working behind the scenes, shaping how your nervous system navigates the world. Its influence is broad, indirect, and constant. Understanding it is less about labeling feelings, and more about recognizing that every mental state arises from a living adaptive system, one that is always adjusting, always responding, and always trying to maintain a workable balance between change and stability. That balance will guide the next part of this exploration, where we move from what serotonin is to how it actually communicates inside the brain, and how a few small clusters of neurons manage to influence almost every corner of your inner world. Serotonin earns its place in the brain not by force but by persistence. It does not arrive in sharp bursts like some other neurotransmitters. It does not announce itself with urgency. Instead, it works in a steady, modulating way, shaping the conditions under which other signals operate. That is why it is often described as a gentle messenger, not because it is weak, but because its influence is distributed and sustained rather than explosive. To understand how this works, it helps to look at how neurons normally communicate. A neuron fires an electrical impulse along its length. When that impulse reaches the end, it triggers the release of a neurotransmitter into the tiny gap between cells, called the synapse. The chemical crosses that gap and binds to receptors on the next neuron. That binding changes how likely the receiving neuron is to fire. In this way, information moves through the brain. Some neurotransmitters act like on-off switches. They either excite or inhibit their targets quickly and decisively. Glutamate, for example, is the main excitatory transmitter. It drives activity. GABA is the main inhibitory one. It suppresses firing. These two form the backbone of moment-to-moment brain activity. Serotonin works differently. Rather than carrying precise messages from one specific neuron to another, many serotonin neurons release their transmitter in a way that affects broader areas. Some of their connections are traditional synapses, but many release serotonin into the surrounding space, influencing whole neighborhoods of cells at once. This is sometimes called volume transmission. Because of this, serotonin is less about transmitting specific information and more about setting the overall responsiveness of circuits. It alters how easily neurons fire, how strongly they react to incoming signals, and how long those reactions last. It changes the gain of the system, the same way adjusting the sensitivity on a microphone changes how loudly everything comes through. This is why serotonin is so deeply involved in regulation. It does not decide what you think. It shapes how intense, persistent, or flexible your thinking becomes. It does not create fear or calm on its own. It influences how readily your brain enters those states and how easily it leaves them. The neurons that produce serotonin are relatively few in number. In the human brain, there are only tens of thousands of them. Yet their influence reaches billions of other neurons. This is possible because of how widely their fibers spread. From the rougher nuclei in the brainstem, their axons fan out through nearly every major brain region. These neurons fire in patterns that reflect your state of wakefulness, stress, and engagement with the environment. During alert wakefulness, many serotonin neurons are active. During deep sleep, especially during REM sleep, their activity drops sharply. This pattern alone hints at their role. Serotonin is linked to stability, to maintaining organized waking states, and to regulating transitions between different modes of consciousness. When you are awake and attentive, serotonin helps maintain a consistent internal environment. It dampens noise in neural circuits. It prevents runaway reactions. It supports the brain's ability to sustain a task or a train of thought without fragmenting. When you fall asleep, serotonin activity decreases. Other systems take over. This shift allows the brain to enter different patterns of activity, including the vivid, disinhibited patterns of dreaming. The quieting of serotonin is part of what allows that change in mode. This does not mean serotonin keeps you awake. It means it participates in the balance between structured waking consciousness and the more fluid states of sleep. It helps define the boundaries. One of the most important roles of serotonin is in the prefrontal cortex. This region sits behind your forehead and is involved in planning, impulse control, emotional regulation, and flexible thinking. It is what allows you to pause, to consider alternatives, to override immediate reactions. Serotonin input to this region helps stabilize its activity. It supports the ability to maintain goals, to inhibit impulsive responses, and to shift perspective. When serotonin signaling here is disrupted, people often experience increased impulsivity, difficulty regulating emotion, and a sense of mental rigidity. This does not manifest as a single feeling. It manifests as patterns. Thoughts may loop more easily. Reactions may feel harder to stop once they start. Small stresses may escalate quickly. It becomes more difficult to step back from an emotional response. In the limbic system, which includes structures like the amygdala and hippocampus, serotonin influences how emotional significance is assigned and how memories are encoded. The amygdala helps detect threat and relevance. The hippocampus helps form new memories and contextualize experience. Serotonin modulates the sensitivity of these structures. It can dampen excessive threat responses. It can influence how strongly emotional experiences are stamped into memory. This is part of why chronic stress, which alters serotonin signaling, can lead to heightened anxiety and persistent negative recall. The hippocampus is also one of the few regions in the adult brain where new neurons continue to be generated. Serotonin influences this process, known as neurogenesis. Adequate serotonin signaling supports the growth and survival of these new cells. This is one reason long-term changes in serotonin availability can affect mood over weeks rather than hours. Structural changes take time. The gentle quality of serotonin's action comes from this combination of breadth and subtlety. It does not shout. It adjusts. It nudges systems towards certain ranges of activity. It stabilizes patterns over time. This is also why changes in serotonin often feel global rather than specific. People do not usually report more serotonin. They report shifts in baseline experience. The world feels slightly more manageable. Thoughts feel less sticky. Emotional reactions feel less extreme. Or, in the other direction, everything feels a bit heavier, more effortful, more prone to spiral. Serotonin's effects are not uniform because its receptors are not uniform. Different receptor types are distributed in different brain regions. Some receptors respond quickly and briefly. Others trigger cascades of intracellular changes that unfold over minutes or hours. For example, some serotonin receptors are ion channels. When serotonin binds, they open and allow charged particles to flow, changing the electrical state of the neuron immediately. Other receptors are G-protein coupled. They do not directly change electrical activity. Instead, they activate signaling pathways inside the cell that alter gene expression, protein synthesis, and receptor sensitivity. This diversity allows serotonin to operate on multiple timescales. It can influence moment-to-moment excitability and longer-term plasticity. It can shape how circuits behave now and how they adapt over time. It also means that the same molecule can have opposite effects in different contexts. In one region, serotonin might quiet activity. In another, it might enhance it. The outcome depends on which receptors are present and how they are coupled to intracellular machinery. This complexity is one reason why early attempts to reduce mood disorders to low serotonin were incomplete. What matters is not just how much serotonin is present, but how it is released, where it acts, which receptors are engaged, and how those receptors are regulated. Stress, for instance, alters serotonin signaling. Acute stress can increase serotonin release in some regions and decrease it in others. Chronic stress can change receptor sensitivity and reduce neurogenesis. These changes do not produce a single uniform effect. They shift the balance of multiple interacting systems. Sleep deprivation also alters serotonin dynamics. When sleep is disrupted, the normal daily rhythm of serotonin neuron activity is disturbed. This affects emotional regulation, attention, and impulse control. Anyone who has been severely sleep deprived recognizes the feeling. Emotions become less stable, reactions more immediate, perspective harder to maintain. Light exposure influences serotonin as well. Bright light, especially in the morning, increases serotonin turnover in the brain. This is one reason light therapy can help with seasonal mood changes. It interacts with the same systems that regulate circadian rhythms and melatonin production. Movement and physical activity also modulate serotonin signaling. Exercise increases the availability of tryptophan in the brain and enhances serotonin synthesis. Over time, it can alter receptor expression and improve regulatory balance. These effects are gradual and cumulative. All of these influences converge on the same theme. Serotonin is not a switch. It is a coordinator. It integrates information about internal state, environmental conditions, and behavioral context. It adjusts how the brain allocates its resources. This coordination is especially important in situations that require flexibility. When circumstances change, the brain must shift strategies. It must let go of one pattern and adopt another. Serotonin facilitates this process. It reduces the tendency for circuits to become locked into repetitive loops. In disorders where serotonin regulation is impaired, rigidity often appears. Thoughts repeat, worries circle, behaviors become habitual even when they are no longer helpful. This is seen in depression, in certain anxiety disorders, and in obsessive patterns. Again, this does not mean serotonin is the sole cause. It is one component of a larger regulatory network, but it plays a central role in how easily the brain can move between states. The gentleness of serotonin is also evident in how its absence is experienced. A sudden drop does not usually produce a dramatic crash. It produces a subtle shift. The mind feels less buffered. Stresses penetrate more deeply. Emotional recovery takes longer. There is a sense of reduced margin. People often describe this as heaviness, fragility, or mental fatigue. It is not a specific emotion. It is a change in how emotions behave. This is why improving serotonin function does not feel like adding happiness. It feels like restoring capacity. The capacity to pause, to reflect, to tolerate discomfort without being overwhelmed, to maintain continuity across changing conditions. From a biological perspective, this makes sense. Organisms that can regulate internal state have an advantage. They can respond to threats without becoming destabilized. They can pursue goals without being derailed by every fluctuation. Serotonin is part of the machinery that enables that stability. It evolved long before humans. Versions of serotonin exist in simple organisms. It regulates movement in worms. It influences feeding in insects. It modulates social behavior in mammals. Its role as a coordinator is ancient. In humans, this ancient system operates within a highly complex brain. It interacts with language, memory, culture, and self-concept. The same molecule that regulates gut motility in simpler creatures now participates in shaping abstract thought and long-term planning. Yet its mode of action remains fundamentally the same. It does not command. It modulates. It biases the system towards certain ranges of behavior. As we continue, we'll look more closely at how serotonin shapes emotional tone specifically, how it influences what we call mood, how it interacts with other systems that generate feeling, and how small shifts in this gentle messenger can change the landscape of experience over days, weeks, and years. The next part will move from the mechanics of signaling to the lived reality of emotion, not by labeling feelings, but by tracing how the brain constructs them and how serotonin participates in that construction. Emotion does not begin as a feeling, it begins as a pattern of activity. Long before you notice sadness, relief, tension, or contentment, your brain is already evaluating information. Sensory input arrives, internal states shift, memories become active, predictions are formed. All of this happens below the level of awareness. What you eventually experience as an emotion is the surface of a much larger process unfolding across many interconnected systems. Serotonin participates in this process not by creating specific emotions, but by shaping the way emotional systems behave. It influences how quickly an emotional response is triggered, how intensely it builds, how long it persists, and how easily it settles. It acts on the machinery that determines whether a reaction feels like a brief ripple or a sustained wave. To understand this, it helps to separate emotion into components. There is detection, the brain's assessment of what is happening. There is valuation, the assignment of meaning or relevance. There is activation, the mobilization of bodily and mental resources. And there is regulation, the process that keeps the whole system from becoming unstable. Serotonin is deeply involved in that last component. It does not tell the brain what to value. It does not decide whether something is good or bad. It influences how strongly those evaluations translate into ongoing mental and physiological states. One of the central emotional structures in the brain is the amygdala. This almond-shaped region plays a key role in detecting significance, especially potential threat. It responds to novelty, uncertainty, and cues associated with danger. When it becomes active, it triggers changes in attention, heart rate, muscle tone, and memory encoding. The amygdala does not operate in isolation. It is connected to the prefrontal cortex, the hippocampus, the hypothalamus, and brainstem nuclei. Together, these regions coordinate emotional responses. Serotonin fibers innervate all of these areas. In the amygdala, serotonin modulates excitability. It can reduce the likelihood that ambiguous stimuli trigger a full threat response. It does not eliminate fear, it adjusts sensitivity. When serotonin signaling is balanced, the amygdala still responds to genuine danger, but it is less likely to overreact to uncertainty or minor stressors. Emotional responses remain proportional. When serotonin regulation is disrupted, this balance shifts. The amygdala can become more reactive. Signals that would normally be dismissed as neutral or manageable begin to register as significant. The threshold for activation lowers. This does not feel like low serotonin. It feels like being on edge. It feels like emotional volume has been turned up. This change in sensitivity has cascading effects. A more reactive amygdala sends stronger signals to other regions. The hypothalamus increases stress hormone release. The brainstem increases arousal. The hippocampus encodes the experience with heightened emotional weight. Over time, these patterns reinforce themselves. The brain becomes tuned to expect threat. Neutral events are interpreted through a lens of caution. Emotional responses begin earlier and last longer. Serotonin's role here is not to suppress emotion, but to maintain a workable dynamic range. It helps prevent the system from saturating. It supports a buffer between stimulus and response. In the prefrontal cortex, serotonin influences a different aspect of emotional life. This region integrates information across time. It allows you to reflect on feelings, to consider context, to inhibit immediate impulses. It is essential for emotional regulation in the everyday sense. Serotonin input to the prefrontal cortex supports this integrative function. It stabilizes activity patterns. It promotes sustained engagement with a goal or perspective. It facilitates cognitive control over emotional impulses. When this system functions well, you can experience an emotion without being consumed by it. You can feel anger without acting on it. You can feel sadness without losing all momentum. You can notice anxiety and still choose how to respond. When serotonin regulation in this region is impaired, that capacity weakens. Emotional responses more easily bypass reflective processes. Reactions feel automatic. Once a feeling arises, it becomes harder to shift away from it. This is not because the emotion is stronger in some absolute sense. It is because the mechanisms that normally modulate and contextualize it are less effective. The emotional signal carries more weight relative to regulatory signals. The hippocampus adds another layer. This structure binds experiences into memory and provides contextual information. It helps distinguish between what is happening now and what happened before. It allows the brain to recognize that a current situation is not the same as a past one. Serotonin influences hippocampal plasticity. It affects how new experiences are encoded and how old memories are retrieved. Balanced serotonin signaling supports flexible memory use. It allows the brain to learn without becoming trapped in past patterns. When this balance shifts, memory can become biased. Negative experiences may be encoded more strongly. Retrieval may favor similar past events. This can create a feedback loop in which present experiences are interpreted through a narrow, emotionally charged lens. This is one way in which serotonin shapes emotional tone over time. It does not merely affect how a single event feels. It influences how experience accumulates and how the past informs the present. Mood, in this context, is not a single feeling. It is a background state. It reflects the average behavior of emotional systems over time. It is the baseline from which specific emotions arise. Serotonin contributes to this baseline by regulating how readily the system enters and exits emotional states. It affects the probability distribution of experiences. With balanced regulation, the system explores a wide range of states and returns to center. With impaired regulation, it becomes biased towards certain regions. In depression, for example, people often describe not just sadness but heaviness, inertia, and loss of flexibility. Emotions do not flow as freely. Positive experiences register less strongly. Negative states linger. This pattern reflects changes in multiple systems. Dopamine signaling related to reward is altered. Stress hormone regulation is disrupted. Inflammatory pathways may be involved. Serotonin participates in these changes by influencing plasticity, reactivity, and regulatory tone. It is important to avoid simplistic narratives. Depression is not caused by low serotonin, in the way anemia is caused by low iron. It is a complex condition involving genetics, development, stress history, and ongoing context. Serotonin is one node in this network. However, because serotonin influences how emotional circuits adapt, small changes in its signaling can have cumulative effects. Over weeks and months, altered serotonin function can reshape how the brain responds to experience. This is why treatments that affect serotonin take time to change mood. Increasing serotonin availability does not immediately produce emotional relief. It initiates gradual adjustments. Receptors adapt. Gene expression changes. New neurons survive in the hippocampus. Circuits slowly reorganize. During this period, people often report subtle shifts before any clear change in mood. Sleep may improve. Appetite may normalize. Emotional reactivity may soften. These are not dramatic, but they change the terrain on which emotions arise. Anxiety provides another lens. In anxiety disorders, the brain's threat detection systems are often hyper-responsive. Uncertainty feels intolerable. Anticipation itself becomes distressing. Serotonin modulates this by influencing how strongly the brain predicts danger and how long those predictions persist. Balanced serotonin signaling allows uncertainty to exist without immediate escalation. It supports a wider margin of safety. When this regulation weakens, the mind fills gaps with worst-case scenarios. Emotional responses are triggered by thoughts rather than events. The system spends more time in a state of readiness. Again, this is not simply too little serotonin, it is altered coordination. The messenger's timing, distribution, and receptor interactions have shifted. One of the subtle but important ways serotonin shapes emotion is through patience. Many studies link serotonin to impulse control and delayed gratification. This is not a moral trait, it is a neurobiological capacity. When serotonin regulation is intact, the brain can tolerate waiting. It can hold a goal across time. It can endure discomfort in service of something larger. This affects emotional life because many difficult feelings involve waiting. Waiting for relief, for change, for understanding. Reduced serotonin function makes waiting harder. The present moment feels more pressing. Discomfort feels more urgent. This increases the emotional weight of frustration and disappointment. This capacity for temporal extension is deeply tied to mood. A system that cannot hold the future easily becomes trapped in the present. If the present feels bad, that trap becomes distressing. Serotonin contributes to the brain's ability to represent time and continuity. It supports the sense that experiences pass, that states change, that what is happening now is not permanent. When this sense weakens, emotional states feel more absolute. Sadness feels endless. Anxiety feels inescapable. These are not philosophical judgments. They are subjective experiences shaped by how neural systems represent time and change. Emotion is also embodied. It involves heart rate, muscle tone, breathing, gut activity. Serotonin influences many of these systems through its peripheral roles and its central connections to autonomic centers. In the brainstem and hypothalamus, serotonin modulates autonomic output. It influences how strongly the body mobilizes in response to emotional signals. Balanced regulation prevents excessive physiological arousal. When this balance is disturbed, emotional states become more physically intense. Anxiety includes palpitations, tightness, nausea. Sadness includes heaviness, fatigue, slowed movement. These sensations feed back into the brain, reinforcing the emotional state. This feedback loop is part of why emotions feel so real. They are not abstract. They are bodily states interpreted by the brain. Serotonin shapes this loop by influencing both ends. It modulates central emotional circuits and peripheral bodily responses. It coordinates mind and body. Over time, patterns of serotonin regulation become part of a person's emotional style. Some people are naturally more reactive. Some recover quickly. Some dwell. These differences arise from genetics, early development, and lived experience. Early life stress, for example, can alter serotonin receptor expression and stress hormone interactions. These changes persist into adulthood. They shape how the brain responds to later challenges. This does not fix destiny. The brain remains plastic. But it means that emotional tendencies have biological substrates. They are not simply habits of thought. Understanding this can change how we relate to emotion. It reframes feelings as processes rather than verdicts. An emotion is not a truth about the world. It is a state generated by a system trying to interpret and respond. Serotonin helps determine how that system behaves. It influences whether emotional signals are brief or prolonged, narrow or flexible, overwhelming or manageable. It does not eliminate pain. It does not guarantee ease. It supports the brain's ability to navigate between states without becoming stuck. As we move forward, we will look more closely at how serotonin interacts with stress systems, and how it contributes to the experience of calm and safety. That interaction reveals why environments, habits, and rhythms matter so much for emotional health, and why small daily factors can accumulate into significant shifts in how life feels. Stress is not a flaw in the human system. It is a built-in response shaped by evolution to protect a living body. Long before modern life introduced deadlines, traffic, and constant notification sounds, stress existed as a survival mechanism. It mobilized energy. It sharpened attention. It prepared muscles to move. It helped organisms respond quickly to danger. At its core, stress is a coordinated shift in the body and brain toward readiness. The heart beats faster. Breathing becomes shallower and quicker. Blood flow shifts toward muscles. Digestion slows. Attention narrows. Thought becomes focused on what might go wrong and how to prevent it. These changes are not random. They are orchestrated by a network involving the hypothalamus, the pituitary gland, the adrenal glands, and multiple brainstem nuclei. This network is often called the stress response system. When activated, it releases hormones such as cortisol and adrenaline. These chemicals act throughout the body and brain. They increase energy availability. They heighten alertness. They bias perception toward threat. In short bursts, this system is useful. It helps you react. It helps you adapt. It helps you survive. The problem arises when stress becomes chronic, when the system is activated not for moments, but for days, weeks, or months. When the brain remains in a state of readiness even in the absence of immediate danger. When the body begins to treat everyday life as if it were an emergency. This is where serotonin becomes especially important. Serotonin interacts with the stress system at multiple levels. It influences how easily the stress response is triggered. It shapes how intensely it unfolds, and it affects how efficiently the system returns to baseline once the trigger has passed. In a balanced state, stress activation rises when needed and falls when no longer necessary. The body mobilizes and then recovers. The brain becomes alert and then relaxes. This rhythm preserves energy and prevents wear. Serotonin supports this rhythm by modulating the sensitivity of stress-related circuits. In the hypothalamus, serotonin influences the release of corticotropin-releasing hormone, which initiates the hormonal cascade of stress. In the brainstem, it affects autonomic output. In limbic regions, it alters how threat is perceived. When serotonin signaling is stable, these systems remain proportionate. The stress response responds to actual challenges. It does not remain chronically engaged. The brain distinguishes between urgency and inconvenience. When serotonin regulation weakens, this balance shifts. Stress circuits become easier to activate and harder to quiet. Cortisol levels may remain elevated. The autonomic nervous system may favor sympathetic activation, the branch associated with arousal and vigilance. This does not feel like a chemical change. It feels like tension. It feels like restlessness. It feels like being unable to fully relax even in safe conditions. It feels like carrying a background sense of unease. The experience of safety is not merely the absence of threat. It is an active physiological state. The body must recognize that it is safe in order to disengage from defense. This recognition depends on sensory input, context, and internal signaling. Serotonin contributes to this recognition. It helps the brain interpret cues of safety. It supports parasympathetic activity, the branch of the autonomic nervous system associated with rest, digestion, and recovery. It facilitates transitions out of heightened arousal. When serotonin function is intact, moments of calm are accessible. The body can downshift, muscles can release, breathing can deepen, thoughts can widen. The system returns to baseline. When serotonin signaling is disrupted, these transitions become less reliable. The stress response lingers. Even in quiet settings, the body remains partially mobilized. The mind continues to scan for problems. Calm becomes harder to reach. This is not a failure of will. It is a shift in regulation. Chronic stress itself alters serotonin function. Elevated cortisol affects serotonin synthesis and receptor sensitivity. Stress hormones influence gene expression in serotonin neurons. Over time, the system adapts to a higher baseline of arousal. This creates a feedback loop. Stress alters serotonin. Altered serotonin reduces the system's ability to disengage from stress. The organism becomes more reactive. Each stressor leaves a deeper imprint. In this state, even small challenges can trigger disproportionate responses. A minor conflict feels overwhelming. A delay feels intolerable. The body reacts as if each event were urgent. Serotonin normally dampens this amplification. It provides a buffer between stimulus and response. It does not eliminate stress, it limits its spread. One way to see this is in startle responses. Studies show that serotonin influences how strongly organisms react to sudden stimuli. With balanced signaling, a sudden noise produces a brief spike in arousal that quickly resolves. With impaired regulation, the same noise produces a larger and more prolonged reaction. This difference is not about bravery or personality. It reflects how efficiently the nervous system resets. Another example lies in rumination. Under stress, the brain tends to replay events, anticipate outcomes, and search for solutions. This can be useful. It becomes maladaptive when it loops without resolution. Serotonin influences the persistence of these loops. Balanced signaling supports cognitive flexibility. It allows thoughts to shift. It prevents the mind from becoming stuck in repetitive patterns. When this regulation is weakened, stress-related thoughts repeat. The brain remains engaged with the same themes. The stress response is reactivated by memory and anticipation, not just by events. This creates the experience of constant mental activity. Even in stillness, the system remains busy. The body cannot fully rest because the brain continues to signal potential threat. Calm in this context is not simply a feeling. It is a physiological state in which stress circuits are quiet and restorative processes dominate. It is associated with lower heart rate, deeper breathing, digestive activity, and a sense of internal space. Serotonin helps make this state accessible. It supports the neural conditions that allow downshifting. It interacts with brainstem nuclei that regulate breathing and heart rate. It influences vagal tone, a measure of parasympathetic activity. Higher vagal tone is associated with greater emotional resilience. It reflects a nervous system that can move between arousal and rest smoothly. Serotonin contributes to this flexibility. This is one reason why practices that enhance parasympathetic activity often interact with serotonin pathways. Regular sleep, exposure to natural light, physical movement, and social connection all influence serotonin regulation. They are not merely psychological interventions, they are physiological inputs. Light, especially morning light, increases serotonin turnover in the brain. It signals the system to align with the external day. This helps regulate circadian rhythms and stress hormone patterns. People who lack sufficient light exposure often experience altered mood and heightened stress reactivity. Sleep allows stress hormones to reset. During certain stages of sleep, serotonin neuron activity decreases, allowing other systems to recalibrate. Chronic sleep deprivation disrupts this rhythm. Stress hormones remain elevated. Serotonin regulation becomes less stable. Physical movement increases tryptophan availability in the brain and enhances serotonin synthesis. It also activates stress systems in a controlled way, followed by recovery. This trains the system to mobilize and relax efficiently. Social interaction influences serotonin through multiple pathways. In mammals, social cues signal safety. Physical presence, vocal tone, and facial expression inform the nervous system that threat is low. Serotonin participates in processing these cues and adjusting arousal accordingly. Isolation, by contrast, deprives the system of safety signals. The brain becomes more vigilant. Stress circuits remain active. Over time, this alters regulatory balance. These effects are not moral, they are biological. The nervous system evolved in environments where light, movement, and social presence were regular. Modern conditions often disrupt these inputs. When people describe feeling on edge all the time, they are often experiencing a nervous system that rarely fully disengages. Serotonin's buffering role is compromised by chronic activation. Medication that alters serotonin signaling can restore some of this buffer. By increasing the availability of serotonin in synapses, these treatments can reduce baseline reactivity. They do not remove stressors. They alter how the system responds. Over weeks, changes in receptor expression and neural plasticity occur. The brain becomes less prone to overactivation. The stress response becomes more proportionate. This is why such treatments often reduce physical symptoms of anxiety before changing mood. Heart rate stabilizes, sleep improves, muscular tension decreases. The body begins to experience moments of safety again. Those moments are essential. They allow recovery. They prevent exhaustion. They create the conditions in which emotional processing can occur without overwhelming the system. Calm is not the absence of challenge. It is the ability to return from challenge. Serotonin supports that return. In its absence, the system behaves as if danger is continuous. Even when nothing is happening, the body remains prepared. This state is costly. It consumes energy. It impairs digestion. It disrupts sleep. It narrows attention. Over time, this state can feel like identity. People begin to describe themselves as anxious or tense, but what they are experiencing is a regulatory pattern that has become habitual. Understanding this reframes the experience. It is not that the world is always threatening. It is that the system is tuned toward threat. Retuning does not require force. It requires inputs that signal safety and stability. It requires time for neural systems to adapt. It requires reducing the constant activation that keeps stress circuits engaged. Serotonin is central to this retuning. It does not create peace. It enables the nervous system to recognize when peace is possible. As this exploration continues, we'll move from stress toward the broader architecture of emotional balance. We'll look at how serotonin contributes not only to the reduction of distress, but to the capacity for positive engagement, motivation, and a sense of forward movement. These aspects are not separate from calm. They arise from the same regulatory foundation. Serotonin does not create happiness in the way a switch creates light. There is no point in the brain where a surge of serotonin produces a clear, identifiable feeling called happiness. What it does instead is shape the conditions under which positive states can arise, persist, and integrate into a person's sense of life. Happiness, in real human terms, is not a single emotion. It is a pattern over time. It is the sense that life is workable, that moments of ease are possible, that effort leads somewhere, that pleasure can be felt without being immediately overshadowed. It is the ability to experience interest, connection, and satisfaction without constant internal resistance. Serotonin contributes to this pattern by influencing three broad dimensions of experience: emotional stability, cognitive flexibility, and temporal continuity. Together, these form what might be called the architecture of well-being. Not joy in isolation, but a structure in which joy can exist. The first of these is emotional stability. This does not mean constant positivity. It means that emotions have boundaries. They rise and fall. They do not flood the system or vanish entirely. They remain in proportion to events. When serotonin regulation is intact, emotional responses are modulated. Negative feelings still occur, but they do not automatically dominate. Positive experiences are able to register. They are not immediately drowned out by internal noise. This stability is not dramatic. It often feels like neutrality with capacity. A person may not feel euphoric, but they can feel okay. They can tolerate minor discomfort. They can experience a pleasant moment without bracing for collapse. In states where serotonin function is impaired, this stability weakens. Emotions become more polarized. Negative states are heavier and more persistent. Positive states are brief or muted. The emotional landscape becomes uneven. This unevenness erodes happiness not by removing pleasure, but by altering its weight. A good moment no longer balances a difficult one. It feels fragile. It disappears quickly. The system returns to baseline in a way that favors heaviness. Serotonin's role here is subtle. It does not generate content. It alters how content is integrated. It affects the baseline against which experiences are measured. The second dimension is cognitive flexibility. Happiness depends on the ability to move between perspectives, to let go of one thought and adopt another, to reinterpret a situation, to imagine alternatives, to shift attention. This flexibility is a property of neural networks. It depends on how easily circuits disengage from one pattern and enter another. Serotonin supports this by modulating the persistence of activity in cortical regions. With balanced signaling, thoughts arise and pass. Attention can be redirected. Setbacks can be contextualized. A person can hold multiple interpretations at once. When this regulation weakens, the mind becomes more rigid. Thoughts repeat. Interpretations narrow. Attention becomes captured by a limited set of themes. This rigidity makes happiness difficult because happiness requires movement. It requires the ability to notice new input and update internal models. A mind that cannot shift easily becomes trapped in its own predictions. It expects disappointment. It anticipates loss. It filters experience through a narrow frame. Even positive events are processed through this filter. Serotonin influences this by affecting prefrontal networks and their interaction with limbic systems. It supports the capacity to override automatic responses and consider alternatives. It does not decide what those alternatives are, it makes them accessible. This is why people often describe recovery from depression not as sudden joy, but as the return of mental space, the return of options, the return of the sense that more than one outcome is possible. The third dimension is temporal continuity. Happiness is not only about what you feel now, it is about how the present connects to the future and the past. It is the sense that time moves and that movement carries meaning. Serotonin contributes to how the brain represents time. It influences patience, anticipation, and the ability to hold goals across delay. It affects whether the future feels reachable or abstract. In a system with intact regulation, the future exists as a real dimension. Effort now is linked to outcome later. Discomfort is framed as temporary. The present is one point in a sequence. When serotonin regulation is impaired, time flattens. The future loses weight. The present feels endless. Discomfort feels permanent. The mind struggles to project beyond the current state. This has profound effects on happiness. If the future does not feel real, motivation collapses. If effort does not connect to outcome, action feels pointless. If change does not feel possible, positive emotion loses its foothold. Serotonin does not create optimism. It supports the neural machinery that allows the future to be represented as a meaningful dimension. It supports the experience of continuity. These three dimensions, stability, flexibility, and temporal continuity, form the scaffolding on which positive states rest. Without them, pleasure becomes fragile. Contentment becomes rare. Engagement feels costly. Happiness in this framework is not a peak. It is a pattern of accessibility. It is the ability to experience a range of states without being dominated by any single one. Serotonin contributes to this accessibility by regulating thresholds. It influences how much input is required to shift state. It affects how easily the system moves. In practical terms, this means that in a well-regulated system, small positive events can matter. A warm drink, a quiet room, a brief conversation. These register. They accumulate. They offset. In a dysregulated system, the same events occur, but they fail to register with the same weight. They feel distant. They slide off. The system remains anchored in a narrow band of experience. This is why people in depressed states often say they know, intellectually, that good things exist but cannot feel them. The sensory input reaches the brain, but the integrative machinery does not assign it lasting significance. Serotonin influences that machinery. It modulates how experience is encoded, how it is integrated into ongoing narrative. This narrative dimension matters. Human well-being is deeply tied to story, not in a literary sense, but in a cognitive one. The brain constantly constructs a model of what is happening, what has happened, and what might happen. Serotonin participates in shaping this model by affecting which signals dominate. It does not write the story. It sets the conditions under which certain themes become persistent. In a balanced system, the narrative is varied. It includes setbacks and progress. It allows for surprise. It updates. In a dysregulated system, the narrative becomes repetitive. It emphasizes threat, loss, or futility. It resists revision. Happiness is not the absence of negative chapters. It is the presence of movement. It is the ability for the story to change. Serotonin's contribution to this change is indirect but central. It supports the brain's capacity to revise its own patterns. This is why interventions that affect serotonin often feel underwhelming at first. People expect a feeling. What they receive is space, a slight reduction in internal pressure, a bit more room between thought and reaction. Over time, this space allows new patterns to form. It allows behavior to shift. It allows experiences to accumulate differently. Happiness, then, emerges not as a chemical effect, but as a consequence of altered dynamics. The system becomes more open, more resilient, more able to engage. This perspective also explains why happiness cannot be installed by chemistry alone. Serotonin creates conditions. It does not fill them. Experiences still matter. Relationships still matter. Activity still matters. The brain still learns from what happens. What serotonin does is determine how the brain learns. It influences whether learning is biased toward threat or toward possibility. It shapes the emotional weighting of memory. A person in a balanced state may experience a setback and encode it as one event among many. A person in a dysregulated state may encode it as confirmation of a global pattern. The difference lies not in intelligence or insight, but in how neural systems integrate information. Understanding this reframes the pursuit of happiness. It is not about maximizing pleasure. It is about maintaining a system capable of integration. Serotonin is one of the regulators of that system. It does not guarantee joy. It makes joy possible. As we continue, we will move from this architectural view towards specific daily rhythms. We will explore how serotonin contributes to the differences between morning and night, between wakefulness and rest, and how these cycles influence emotional tone across a day. These rhythms are not separate from happiness, they are part of its foundation. Morning and night are not just different times of day, they are different biological states. The body you inhabit at sunrise is not the same body you inhabit at midnight, even if nothing visible has changed. Inside, hormone levels shift, neural activity patterns reorganize, temperature rises and falls, and entire networks move into different operating modes. These changes are not psychological habits. They are built into the structure of your nervous system. Serotonin sits at the center of this daily transformation. The brain runs on rhythms. The most prominent is the circadian rhythm, a roughly 24-hour cycle governed by a small cluster of neurons in the hypothalamus called the suprachiasmatic nucleus. This region receives direct input from the eyes. It does not process images, it measures light. When daylight reaches the retina, specialized cells signal the clock. That signal tells the brain that a new day has begun. From there, the message spreads. Hormone release shifts. Body temperature begins to rise. Cortisol increases in the early morning, helping mobilize energy. Melatonin production, which promotes sleep, shuts down, and serotonin activity changes. Serotonin neurons increase their firing during waking hours. This does not create alertness on its own, but it supports the transition into a stable waking state. It helps coordinate the shift from internal, sleep-oriented processing to outward engagement. Morning often feels clearer not because problems disappear, but because the system that regulates attention and emotional tone is more organized. Serotonin contributes to this organization. It stabilizes cortical activity. It supports goal-directed behavior. It reduces the background noise that accumulates during fatigue. This is why many people feel more capable in the morning. Thoughts line up more easily. Tasks feel more manageable. Emotional reactions are often less intense. The brain is operating in a mode designed for interaction with the environment. As the day progresses, this state gradually changes. Serotonin activity remains elevated through much of the daylight period, but other systems begin to shift. Adenosine accumulates in the brain, increasing sleep pressure. Body temperature peaks and then slowly declines. Melatonin production begins to rise in the evening as light exposure decreases. Serotonin and melatonin are biochemically linked. Melatonin is synthesized from serotonin. As evening approaches, some serotonin in the pineal gland is converted into melatonin. This does not deplete brain serotonin directly, but it reflects a coordinated transition. The system begins to favor rest over engagement. At the same time, serotonin neuron firing gradually decreases. The tone of the brain changes. Circuits that support sustained attention and impulse control become less active. Emotional regulation becomes more effortful. The boundary between thought and feeling thins. This shift is subtle at first. It appears as mental fatigue. Tasks require more energy. Small irritations feel heavier. Perspective narrows. By late evening, the same thoughts that felt manageable in the morning can feel more absolute. This is not because circumstances changed. It is because the regulatory machinery has shifted. At night, the brain becomes more internally oriented. Sensory input is reduced. External demands fade. Memory and emotion take on more weight. This is adaptive. Night is a time for integration. The brain reviews experience. It consolidates memory. It reorganizes. But this internal orientation also means that unresolved material becomes more prominent. Worries surface, regrets replay, anticipation becomes vivid. Without the stabilizing influence of daytime serotonin activity, these processes feel stronger. The prefrontal cortex, which supports perspective and inhibition, is less active in the late evening. Serotonin's modulatory influence there is reduced. This makes it harder to redirect thought. Loops persist. Emotional content feels closer. This is why people often report that anxiety, sadness, or rumination intensify at night. The same mind in the morning can think about a problem analytically. At night, it feels enveloping. This pattern is not a personal flaw. It is a consequence of circadian neurobiology. The stress system also behaves differently across the day. Cortisol follows a daily rhythm, peaking in the early morning and declining through the evening. This morning peak is part of what supports wakefulness and energy. It also interacts with serotonin systems. In the morning, higher cortisol and higher serotonin activity work together to promote engagement. The system is mobilized but organized. In the evening, cortisol drops and serotonin activity declines. The system becomes less mobilized and less regulated. This combination creates a vulnerability window. Emotional signals that would be dampened earlier in the day carry more weight. The mind becomes more reactive to its own contents. Sleep pressure adds another layer. As adenosine accumulates, neural efficiency decreases. The brain has fewer resources for regulation. Tasks that require effortful control become harder. Emotional filtering weakens. In this state, small triggers can produce disproportionate reactions. A thought about the future can feel urgent. A memory can feel unresolved. The system lacks the buffering capacity present earlier. Serotonin normally contributes to that buffer. It supports the brain's ability to hold multiple perspectives and maintain a sense of proportion. When its activity decreases, that support weakens. This does not mean night is inherently negative. For many, it is a time of creativity, reflection, and quiet. But it is a different mode. It favours depth over breadth. It favours association over control. Dreaming reflects this mode. During REM sleep, serotonin neuron firing drops almost completely. The brain becomes highly associative. Emotional memory is active. Logical constraint is reduced. Experience becomes fluid. The transition into sleep begins before you are aware of it. As evening progresses, the brain shifts toward this internal associative mode. Serotonin's gentle influence fades. Other systems take precedence. This shift explains why decisions made at night often feel different in the morning. Emotional weight changes. Problems that seemed unsolvable regain proportion. The brain has returned to a state with greater regulatory capacity. Understanding this pattern has practical implications. It explains why late-night thinking is rarely productive. It explains why emotional conversations can escalate in the evening. It explains why routines that promote wind-down are effective. Light exposure plays a central role. Artificial light, especially blue-rich light from screens, interferes with melatonin production and circadian signaling. It delays the evening transition. This creates a mismatch. The brain remains partially in daytime mode while other systems move toward night. This mismatch disrupts serotonin rhythms. Neurons receive conflicting signals. The result is often a state of tired alertness. The mind feels wired but unfocused. Emotional regulation is impaired. Sleep onset is delayed. Over time, this pattern erodes the distinction between day and night. The circadian system loses clarity. Serotonin and melatonin rhythms flatten. Mood becomes less stable. Restoring rhythmicity helps restore regulation. Consistent light exposure in the morning strengthens the daytime serotonin pattern. Reduced light in the evening allows melatonin to rise and serotonin activity to fall in a coordinated way. Movement early in the day reinforces this signal. Physical activity increases serotonin synthesis and promotes alignment with circadian timing. The system learns when to engage and when to disengage. Sleep quality depends on this coordination. When serotonin activity decreases smoothly and melatonin rises, the brain transitions into sleep more easily. Emotional processing during sleep becomes more effective. Stress hormone levels reset. The next morning, serotonin activity rises again. The brain re-enters a regulated waking state. Emotional tone resets. Disruptions to this cycle accumulate. Shift work, irregular sleep schedules, and chronic late-night screen use blur the boundaries. The system loses its clear phases. Serotonin regulation becomes inconsistent. Mood follows. Many mood disorders involve altered circadian rhythms. Depression often includes flattened daily variation. Mornings may feel heavy. Evenings may bring agitation. The normal rise and fall of regulatory tone is distorted. Treatments that address mood often address rhythm as well. Light therapy, sleep hygiene, and regular schedules all act on the same underlying systems. They restore timing. They allow serotonin to perform its daily role. Mornings and nights feel different because the brain is different. The chemical environment changes. Neural priorities shift. Serotonin is one of the conductors of this daily transition. The morning brain is outward-facing, structured and buffered. The night brain is inward-facing, associative and less constrained. Neither is wrong. Each serves a purpose. Difficulties arise when the system loses its contrast, when night intrudes into day or day into night, when serotonin's rhythmic modulation becomes irregular. The goal is not to force constant brightness or constant calm. It is to allow the system to move between modes, to engage and disengage, to mobilize and rest. Serotonin supports that movement. It is part of how the brain knows when to be steady and when to loosen its grip. As this exploration continues, The next part will move beyond daily rhythm into the wider body. It will trace how serotonin operates not only in the brain, but throughout the digestive system and peripheral nerves, shaping experience from the inside out in ways that are often felt but rarely understood. Most people imagine serotonin as something that lives in the brain. They picture it floating between neurons, quietly shaping mood and thought. What is far less commonly known is that the vast majority of serotonin in your body is not in your head at all. It is in your gut. Roughly 90% of the body's serotonin is produced in the digestive tract. Specialized cells in the lining of the intestines synthesize it from tryptophan, an amino acid obtained from food. This serotonin never crosses into the brain. The blood-brain barrier prevents it, yet its influence on how you feel is profound. The digestive system is not a passive tube. It is a densely innervated organ, packed with neurons, immune cells, and chemical sensors. It has its own nervous system, often called the enteric nervous system. This network contains hundreds of millions of neurons. It can operate independently of the brain, coordinating digestion, absorption, and movement. Serotonin is one of its primary signaling molecules. In the gut, serotonin regulates the rhythmic contractions that move food forward. It controls how quickly contents pass. It influences secretion, blood flow, and sensitivity. When serotonin release is well coordinated, digestion proceeds smoothly. When it is dysregulated, discomfort appears, cramping, urgency, sluggishness, or pain. But the gut is not isolated. It communicates constantly with the brain through the vagus nerve and through hormonal and immune pathways. Signals from the digestive tract inform the brain about nutrient status, inflammation, and internal conditions. The brain, in turn, adjusts appetite, mood, and energy. This two-way communication is often referred to as the gut-brain axis. Serotonin is a central language in this dialogue. When the gut releases serotonin, it does more than move food. It activates sensory fibers that project to the brainstem. These fibers do not convey words or images, they convey state. They tell the brain whether the internal environment is calm or disturbed. Under ordinary conditions, this signaling remains in the background. You do not feel digestion happening. The system runs quietly. Under stress, however, this balance changes. Stress alters gut motility, blood flow, and immune activity. It also alters serotonin release in the intestines. Many people notice that anxiety produces immediate digestive effects: tightness, nausea, urgency, loss of appetite. These are not symbolic reactions. They are direct consequences of altered signaling. The brain interprets these bodily signals as part of emotional experience. A racing heart, shallow breathing, and a tight stomach combine into a coherent state. The mind labels that state as anxiety or distress. Because gut serotonin shapes these bodily sensations, it indirectly shapes emotion. Chronic stress can sensitize the digestive system. Inflammatory processes alter serotonin handling. Receptors change. Motility becomes irregular. Conditions such as irritable bowel syndrome illustrate this interaction. They involve altered gut signaling and heightened sensitivity, often accompanied by anxiety or depression. In these states, the gut sends frequent signals of discomfort. The brain receives a steady stream of internal disturbance. Even in the absence of external threat, the nervous system remains engaged. The experience becomes one of persistent unease. This is one reason why digestive disorders and mood disorders often coexist. It is not that one causes the other in a simple chain. It is that they share regulatory pathways. The same systems that govern emotional tone govern visceral sensation. Serotonin links them. The gut also hosts a vast population of microorganisms. The microbiome interacts with serotonin in multiple ways. Some bacteria influence tryptophan metabolism. Others produce metabolites that affect enterochromaffin cells, altering serotonin release. Changes in microbial composition can shift gut signaling. Diet, antibiotics, illness, and stress all influence this ecosystem. These shifts modify the chemical messages sent to the brain. Research in this area is still developing, but one pattern is clear. The state of the gut influences the state of the mind. Not metaphorically, physiologically. This does not mean that eating a certain food will instantly change mood. The system operates over time. It is about tone, not spikes. A digestive system that is inflamed, irregular, or deprived of diverse nutrients sends different signals than one that is stable and well supported. Those signals shape baseline arousal and emotional bias. Serotonin's peripheral role also affects energy. The gut determines how nutrients are absorbed. It influences glucose availability. It interacts with insulin. These metabolic signals feed into the brain's sense of capacity. Fatigue, heaviness, and mental fog often follow digestive disruption. They are not separate phenomena. They reflect a system struggling to maintain internal equilibrium. The vagus nerve is a major conduit in this relationship. It carries information from the gut to the brainstem. This information shapes autonomic tone. It influences heart rate, breathing, and inflammatory response. Serotonin modulates vagal signaling. It affects how strongly gut activity is translated into central nervous system response. High vagal tone is associated with resilience. It reflects a system capable of integrating bodily signals without becoming destabilized. Serotonin contributes to this integration. When gut signaling becomes erratic, vagal tone often decreases. The system becomes more reactive. Internal sensations feel more intrusive. The boundary between body and mind blurs. This is the origin of what people call a gut feeling. It is not intuition in a mystical sense. It is the brain interpreting visceral signals. Sometimes those signals correspond to meaningful patterns. Sometimes they reflect internal disturbance rather than external reality. The brain does not distinguish automatically. It constructs meaning from input. Serotonin shapes the quality of that input. This understanding reframes many everyday experiences: the heavy feeling after poor sleep and rushed meals, the lightness after a calm breakfast in sunlight, the tension that accompanies prolonged worry. These are not psychological artifacts. They are physiological states. Serotonin's role beyond the brain reminds us that emotion is embodied. Mood is not confined to thought. It arises from the entire organism. Care for emotional balance therefore includes care for the body's internal environment. Regular meals, hydration, movement, and rest are not lifestyle advice in a moral sense. They are regulatory inputs. They stabilize the gut. They stabilize serotonin signaling. They stabilize the messages the body sends to the brain. This does not guarantee contentment. It reduces background noise. A nervous system receiving steady, non-alarming internal signals is more capable of perspective. It can respond to external events without being overwhelmed by internal disturbance. As we continue, we will move from this bodily foundation back into the mind. We will look at how serotonin connects gut, brain, and memory, shaping motivation and the sense of forward movement. The same molecule that moves food also influences whether effort feels worthwhile. Memory is not a storage cabinet. It is an active process. Every time you remember something, the brain reconstructs it. Circuits light up. Sensory fragments are reassembled. Emotional tone is restored. The past is not retrieved like a file. It is recreated in the present. Serotonin participates in this reconstruction. It influences which memories are likely to surface, how vividly they are experienced, and how strongly they shape current thought. It does this by modulating activity in the hippocampus, the amygdala, and the networks that bind memory to emotion. The hippocampus is essential for forming new memories and for placing experiences in context. It helps distinguish what is happening now from what happened before. It allows the brain to recognize that a present moment is new, even if it resembles something in the past. Serotonin supports the hippocampus in two ways. It influences synaptic plasticity. the process by which connections between neurons strengthen or weaken, and it promotes the survival of newly generated neurons in this region. These new neurons are especially important for pattern separation, the ability to tell similar experiences apart. When this system functions well, the brain can learn without becoming trapped. It can encode experience while maintaining flexibility. A setback today does not overwrite all past success. A difficult conversation does not become a template for every future interaction. When serotonin regulation in this system weakens, this balance shifts. Memory becomes less differentiated. Similar experiences blur. Emotional weight carries forward more easily. The brain becomes more likely to interpret new events through the lens of old ones. This does not feel like a memory problem. It feels like inevitability. It feels like patterns repeating. It feels like the future is constrained by the past. This constriction has direct consequences for motivation. Motivation is not simply the desire to act. It is the brain's calculation that action will lead to meaningful change. It depends on the ability to represent a future that differs from the present and to believe that effort can bridge that gap. Serotonin contributes to this calculation by shaping how the brain links present action to future outcome. It influences how reward circuits interact with planning circuits. It modulates the persistence of goal representations. Dopamine is often described as the neurotransmitter of motivation, but dopamine does not operate in isolation. It signals salience and prediction error. It tells the brain that something is worth pursuing or updating. Serotonin shapes how those signals are integrated over time. It affects whether the system becomes impatient or perseverant. It influences whether a delayed reward still feels real. In a balanced system, effort feels connected to outcome. The brain can hold a goal across time. Temporary discomfort is framed as part of a process. Motivation emerges as a steady orientation toward change. When serotonin regulation weakens, this temporal bridge erodes. The future loses emotional weight. Effort feels disconnected from outcome. Action feels empty. This is one of the central features of depression. It is not merely sadness, it is a collapse of forward momentum. Tasks feel pointless. Goals feel abstract. The system cannot generate a sense that change is possible. This is not laziness. It is a failure of neural representation. Serotonin's role in this failure is indirect but crucial. By altering hippocampal plasticity, prefrontal stability, and limbic reactivity, it changes how experience is integrated into narrative. A mind that cannot update its model of the future becomes stuck. It repeats. It anticipates the same outcome. It conserves energy by disengaging. This disengagement feels like apathy. It feels like exhaustion. It feels like nothing is worth the effort. The same mechanism operates in milder forms. After repeated stress, people often report feeling worn down. They still care, but action feels heavier. Each task requires more activation. The system has learned that effort often leads to strain. Serotonin normally counterbalances this by supporting adaptive learning. It allows negative experiences to be encoded without becoming global. It prevents each difficulty from becoming a rule. Memory and motivation are therefore inseparable. What you remember shapes what you expect. What you expect shapes what you are willing to do. Serotonin influences this loop. In anxiety, the loop takes a different form. Instead of disengagement, there is hyper-engagement. The future feels too real. Possibilities multiply. Threat becomes probable. Here, serotonin's regulatory role is again evident. Balanced signaling limits the spread of anticipation. It prevents the mind from simulating every possible outcome with equal intensity. When this regulation weakens, imagination becomes a source of stress. The brain generates scenarios faster than they can be evaluated. The system becomes saturated with potential futures. Motivation in this state becomes avoidance. Action is driven by the need to reduce uncertainty. Effort is expended not toward growth, but toward control. Both patterns, Disengagement and hypervigilance reflect imbalances in how memory and future representation interact. Both involve altered serotonin regulation. Mental balance lies between these extremes. It is the ability to remember without being trapped and to anticipate without being overwhelmed. Serotonin supports this balance by modulating the persistence of neural activity. It affects how long a memory trace remains active. It influences how strongly an imagined future recruits emotional circuits. With balanced signaling, the brain can let go. It can allow a thought to pass. It can shift focus. It can hold a plan lightly. This lightness is not superficial, it is functional. It allows the system to explore options without committing prematurely. It allows learning without fixation. One way to observe this is in problem solving. In a regulated state, the brain can try an approach, evaluate, and adjust. In a dysregulated state, it either perseverates on one solution or avoids the problem entirely. These patterns are not traits. They are states of a system. Serotonin contributes to the state by altering how readily circuits disengage. It supports transitions. Transitions are essential to mental health. The ability to move from thought to thought, from task to rest, from effort to recovery. Memory, in this context, is not merely recall. It is the ongoing updating of internal models. Motivation is not drive. It is the sense that updating is worthwhile. Serotonin sits at the interface of these processes. This interface is visible in everyday experience. Consider the difference between remembering a mistake as information and remembering it as identity. In one case, the memory informs adjustment. In the other, it defines self. The difference lies in integration. A memory that is integrated remains specific. A memory that dominates becomes global. Serotonin influences this by shaping how emotional weight is distributed. It does not erase negative memory. It prevents it from overwhelming the narrative. When this function is compromised, people often describe feeling defined by past events. They cannot move beyond them. The brain replays the same scenes. The future becomes an extension of the past. Recovery involves restoring the brain's capacity to update, to allow new experience to matter. This is why therapy and medication often work together. Therapy provides new experience and new interpretation. Medication alters the regulatory environment so that these experiences can be integrated. Without regulatory support, new experience may fail to penetrate. Without new experience, regulatory changes may feel empty. Serotonin does not supply meaning. It allows meaning to change. Motivation returns not as enthusiasm, but as possibility. A person may not suddenly want everything. They begin to sense that something might be worth doing. That sense is fragile. It depends on continued integration. It depends on sleep, rhythm, and bodily state. It depends on the system remaining flexible. Memory, motivation, and balance are therefore not separate domains. They are expressions of one regulatory process. Serotonin is one of its central modulators. It allows the brain to remember without freezing, to anticipate without spiraling, and to act without exhausting itself. In the next part of this exploration, we will turn toward what happens when the system drifts out of tune. We will look at how imbalances in serotonin-related regulation manifest as recognizable patterns of suffering, and how those patterns emerge not from a single failure, but from shifts in the way the system holds itself together. When a musical instrument drifts out of tune, it does not stop producing sound. It continues to play. The notes are still there. What changes is how they fit together. Some tones become sharp, others fall flat. Harmony gives way to strain. The music does not disappear, but it becomes harder to listen to. Something similar happens when serotonin regulation begins to drift. Nothing shuts down. The brain still thinks. Emotions still arise. Memory still functions. Motivation still exists in some form. But the coordination between these systems weakens. Signals arrive with altered weight. Responses linger too long or fade too quickly. The mind keeps working, yet the experience of being inside it changes. This drift rarely announces itself in a single moment. It unfolds gradually. The system adapts to stress, sleep loss, irregular rhythm, illness, or prolonged uncertainty. Each of these alters how serotonin neurons fire, how receptors respond, and how circuits integrate information. Over time, the baseline shifts. At first, the changes are subtle. Irritation appears more easily. Recovery takes longer. A poor night of sleep affects the whole day. Thoughts repeat a little more often. The margin between stimulus and response narrows. The person still recognizes themselves. Life still moves. But the system feels less forgiving. As this drift continues, patterns emerge. Emotional responses become less proportional. Minor stressors trigger outsized reactions. Setbacks carry more weight than before. The brain begins to favor certain interpretations, often those associated with threat or futility. This is not because the world has suddenly become more dangerous or meaningless. It is because the regulatory background has changed. Serotonin normally helps distribute emotional weight. It prevents any single signal from dominating. When its modulatory influence weakens, certain circuits gain relative power. Threat detection becomes louder. Negative memory becomes more accessible. The future becomes hard to represent. This imbalance can take different forms. In some people, the system becomes biased toward disengagement. Energy drops. Interest fades. Tasks feel heavy. Pleasure registers weakly. The future feels abstract. This is the terrain of depressive experience. In others, the system becomes biased toward vigilance. The mind scans constantly. Thoughts anticipate problems. The body remains in a state of readiness. Rest feels unsafe. This is the terrain of chronic anxiety. Often, these patterns coexist. Periods of agitation alternate with collapse. The system swings between overactivation and withdrawal. These are not separate disorders in a mechanical sense. They are expressions of how a regulatory system responds to sustained strain. Serotonin's role in these patterns is not that of a missing ingredient. It is the shifting of balance. Receptors change sensitivity. Firing patterns alter. Interactions with stress hormones become maladaptive. Chronic stress is a powerful driver of this drift. Elevated cortisol affects serotonin synthesis. It alters receptor expression. It reduces neurogenesis in the hippocampus. It strengthens amygdala reactivity. Each of these changes biases the system toward threat and rigidity. Sleep disruption compounds the effect. Without consistent rhythm, serotonin neuron activity becomes irregular. Circadian coordination weakens. The daily reset that normally restores balance becomes incomplete. Inflammation also plays a role. Immune signals interact with serotonin pathways. They alter tryptophan metabolism, shifting it away from serotonin synthesis toward other pathways. This reduces availability and changes signaling dynamics. These biological shifts do not occur in isolation. They interact with experience. A person under prolonged stress may encounter setbacks. Those setbacks are encoded by a system already biased toward negativity. The memories carry more weight. Expectations narrow. Motivation drops. The individual may conclude that they are failing, that life has changed, that they are different. From the inside, it feels personal. From the outside, it looks like a change in mood or behavior. From a system perspective, it is a regulatory drift. This drift affects perception itself. The brain does not receive raw reality. It constructs experience. It filters input. It highlights certain features and dampens others. When serotonin regulation is altered, this filtering changes. The mind becomes tuned towards certain classes of information. In depression, neutral stimuli may feel empty. Positive stimuli may fail to register. In anxiety, ambiguous stimuli may feel threatening. The person does not choose this tuning. It emerges from altered circuitry. This is why reassurance often fails. Telling someone that things are not so bad does not recalibrate their system. Their brain is not misinformed. It is operating with a different baseline. The same event is processed differently. The same thought carries more weight. The same memory feels more final. The drift also affects the body. Autonomic tone shifts. Muscles remain tense. Digestion becomes irregular. Sleep becomes shallow or fragmented. These bodily states feed back into the brain, reinforcing the emotional pattern. A person may feel constantly tired, yet unable to rest, or restless, yet unable to act. These are not contradictions. They reflect a system stuck between modes. Serotonin normally supports transitions. It helps the brain move between activation and rest, between focus and release. When this function weakens, transitions become abrupt or incomplete. The system may remain partially activated even in quiet conditions, or it may collapse into disengagement even when engagement is required. These states are often described in moral language: lazy, overreactive, unmotivated, too sensitive, but they are not traits. They are expressions of a regulatory pattern. This pattern can persist even after external stressors resolve. The system learns. It adapts to strain. It does not automatically return. Neural circuits strengthen based on use. A brain that has spent months in vigilance becomes efficient at vigilance. A brain that has spent months disengaged becomes efficient at withdrawal. Serotonin participates in this learning. It influences which patterns are reinforced. This does not mean the system is broken. It means it has adapted to conditions that may no longer be present. Retuning requires time. It requires altering inputs. It requires restoring rhythm. It requires reducing chronic activation. It often requires external support. Medication can help by shifting serotonin dynamics. By increasing availability, it can reduce baseline reactivity. Over weeks, it can promote plasticity. It can allow circuits to weaken maladaptive patterns and strengthen alternative ones. This process is gradual. The brain does not snap back. It reorganizes. Psychological interventions also act on this system. New experiences provide new input. They challenge existing patterns. But without regulatory support, those inputs may fail to integrate. The drift out of tune is therefore not a single event. It is a trajectory. It begins with strain. It becomes habit. It solidifies as expectation. Understanding this changes how we view emotional suffering. It is not a flaw in character. It is a system doing what systems do under prolonged load. Serotonin's role in this is not dramatic. It is infrastructural. It shapes the background against which everything else unfolds. When that background shifts, the world feels different. In the next part, we will look more closely at how this drift appears in the experiences people recognize as depression and anxiety, and why these conditions feel so comprehensive, touching thought, body, and sense of self at once. Depression and anxiety are often described in emotional terms: sadness, fear, heaviness, worry, emptiness. These words capture how the experience feels from the inside, but they do not explain why it feels that way. They describe the surface of a much deeper process unfolding inside the nervous system. Beneath these experiences lies a language the brain uses to coordinate itself. That language is chemical. Neurons do not speak in sentences. They communicate through electrical impulses and chemical messengers. Every thought, every memory, every emotional shift corresponds to changes in this internal dialogue. When that dialogue becomes unbalanced, experience changes. Not symbolically, mechanically. Depression and anxiety are not simply emotional reactions to life. They are states of a biological system that has shifted its operating mode. Serotonin is one of the central participants in that shift. This does not mean that depression is low serotonin, or that anxiety is not enough happiness chemical. Those phrases oversimplify a complex system. but serotonin is deeply involved in the regulation that breaks down in these conditions. In depression, the brain's ability to generate and sustain forward motion weakens. The future feels distant. Effort feels disconnected from outcome. Emotional tone becomes heavy and persistent. Positive experiences register weakly or not at all. From the inside, this feels like a change in self. People often say, "I'm not myself anymore." They may remember caring about things. They may remember energy. But those states feel unreachable. From the perspective of neural systems, several things are happening at once. The hippocampus, which helps contextualize experience and form new memory, becomes less plastic. Neurogenesis slows. Pattern separation weakens. New experiences are less likely to reshape internal models. The prefrontal cortex, which supports planning and perspective, becomes less effective at modulating limbic activity. Thoughts become more rigid. Rumination increases. The amygdala and related structures may remain active, biasing attention toward negative or threatening information. The stress system may remain partially engaged, maintaining elevated cortisol levels that further impair plasticity and regulation. Serotonin interacts with all of these systems. Altered serotonin signaling contributes to reduced plasticity, increased reactivity, and weakened regulatory control. The result is a brain that is less able to update. It continues to operate on models built under strain. It predicts continuation of the current state. It treats heaviness as permanent. This is why depressed thinking often feels absolute. It will always be this way. Nothing helps. There's no point. These are not philosophical positions. They're outputs of a system that cannot easily represent change. Anxiety reflects a different but related shift. Here the system becomes biased toward anticipation. The future feels too present. Possibilities multiply. Uncertainty becomes intolerable. The body remains in a state of readiness. The amygdala and related threat detection circuits are more reactive. The prefrontal cortex struggles to inhibit them. Thoughts about what might happen activate the same systems as actual danger. Serotonin normally dampens this amplification. It limits how far and how long threat signals propagate. When its modulatory influence weakens, imagination becomes a source of stress. The person may know intellectually that nothing is wrong, yet feel as if something is. The body reacts before thought can intervene. Reassurance fails because the system generating the sensation is operating independently of conscious belief. This is why anxiety often feels irrational. The mind and body are not aligned. The body behaves as if danger is present. The mind struggles to explain why. Both depression and anxiety involve changes in how the brain interprets information. They are not simply emotions, they are altered states of perception, memory, and prediction. Chemistry provides the medium for these changes. Serotonin's role is not to dictate content, but to set tone. It influences how signals are weighted, how long they persist, and how easily they are overridden. In depression, reduced regulatory tone allows negative signals to dominate. In anxiety, it allows threat signals to spread. These patterns become self-reinforcing. A depressed system encounters difficulty, encodes it strongly, and updates its model toward hopelessness. An anxious system anticipates danger, reacts strongly, and confirms its expectation. The brain learns from what it experiences. When experience is filtered through altered chemistry, learning becomes biased. This is why these conditions feel so comprehensive. They affect thought, body, sleep, appetite, and identity. They are not confined to mood. A person with depression does not merely feel sad. They may feel slowed, foggy, heavy, disconnected. A person with anxiety does not merely worry. They may feel restless, tense, exhausted, unable to settle. These are whole system states. Language often fails to capture this. People say, "I'm anxious," or "I'm depressed," as if these were simple descriptors. In reality, they are shorthand for a global shift in how the organism operates. Understanding this reduces moral judgment. These states are not weaknesses. They are patterns of regulation shaped by biology and experience. Serotonin is not the sole cause, but it is a major channel through which these patterns express themselves. This is why treatments that alter serotonin can change experience even when circumstances remain the same. The external world does not shift. The internal processing does. Medication does not give a person new beliefs. It changes how strongly existing beliefs are weighted. It changes how easily thoughts disengage. It changes how persistent emotional signals feel. This often appears first in the body. Sleep improves, appetite normalizes, physical tension decreases. These changes signal that autonomic regulation is shifting. Only later does mood change. The person may notice that thoughts no longer loop as tightly, that a problem feels slightly less final, that a pause exists where none did before. These are not dramatic moments, they are small shifts in regulatory capacity. Psychological interventions work on a different layer of the same system. They provide new patterns of experience, they challenge rigid models, they offer alternative interpretations, but without chemical support, those new patterns may fail to integrate. The brain may be too rigid or too reactive to absorb them. Conversely, chemical changes without new experience may feel empty. The system becomes more flexible, but nothing new enters. This is why combined approaches are often effective. Chemistry alters the conditions, experience fills them. The language of chemistry does not replace the language of feeling, it explains it. It tells us that sadness can be a state of neural persistence, that fear can be a state of amplified prediction, that hopelessness can be a failure of future representation. These are not metaphors, they are descriptions of how a system behaves. Serotonin is part of the grammar of that system. It does not speak in words, it adjusts thresholds, it modulates flow, it influences whether a signal becomes a whisper or an echo. Depression and anxiety arise when the system's grammar changes. They are not foreign invaders. They are modes the brain enters under certain conditions. Recovery is not about erasing those modes. It is about restoring range. A healthy system can enter sadness and leave it. It can feel fear and settle. It can engage and rest. It can hold multiple futures in mind. Serotonin supports that range. It does not eliminate pain. It allows movement. As this exploration continues, we will turn toward the everyday factors that influence this chemistry. We will look at how light, sleep, and natural rhythms shape serotonin's behavior, and how small environmental changes can accumulate into large shifts in emotional tone over time. Serotonin does not operate in isolation. It is constantly responding to signals from the world outside your body. Light, darkness, movement, temperature, sound, and rhythm all shape how this system behaves. Long before humans built cities and schedules, these signals were the primary organizers of internal life. They still are. The brain evolved in an environment where light meant day, darkness meant night, movement meant survival, and stillness meant rest. Serotonin became one of the chemicals that translated these environmental patterns into internal states. Light is the most powerful of these signals. Specialized cells in the retina detect brightness, not images. They send that information directly to the brain's central clock in the hypothalamus. From there, timing signals spread through the nervous system. Hormones shift, temperature changes, neural firing patterns adjust. Serotonin neurons respond to this rhythm. During daylight, especially in the morning, serotonin activity increases. This does not create happiness. It creates readiness. It stabilizes waking consciousness. It supports engagement. Morning light is especially influential. Exposure to bright light early in the day strengthens the amplitude of the circadian rhythm. It tells the system clearly when day begins. This sharpens the contrast between day and night. When this contrast is strong, serotonin and melatonin follow clear patterns. Serotonin supports daytime regulation. Melatonin supports nighttime rest. The system moves between modes smoothly. When this contrast weakens, rhythm blurs. Late nights, dim days, and irregular schedules flatten the cycle. Serotonin activity becomes less distinct across time. The brain loses a reliable signal for when to engage and when to release. This blurring has emotional consequences. Mood becomes less stable. Energy fluctuates unpredictably. The system drifts. Seasonal changes illustrate this clearly. In winter, reduced daylight leads to lower serotonin turnover in some individuals. The circadian signal weakens. Melatonin may rise earlier and persist longer. People feel heavier, slower, less motivated. This is not imagination. It is physiology. Light therapy works by restoring that signal. Bright light in the morning increases serotonin activity and realigns circadian timing. Over days and weeks, mood improves. Sleep is the partner of light in this system. During sleep, especially deep and REM sleep, serotonin neuron activity changes dramatically. In REM sleep, it nearly stops. This allows other systems to dominate. Memory is reorganized. Emotional material is processed in a different mode. Healthy sleep is not just rest. It is active recalibration. Stress hormones decline. Autonomic tone shifts toward recovery. Inflammatory signals are modulated. The brain resets. When sleep is shortened or fragmented, this recalibration is incomplete. Stress systems remain partially active. Serotonin rhythms are disrupted. The next day begins without a full reset. Over time, this accumulates. Emotional regulation weakens. Reactivity increases. Thoughts become less flexible. People often underestimate the emotional cost of poor sleep. They interpret irritability, low mood, or anxiety as psychological. In many cases, they are physiological consequences of a system that has not completed its nightly transition. Serotonin sits in the middle of this. It helps define waking tone. It must decrease for sleep to unfold properly. It must rise again for day to feel coherent. Irregular sleep traps the system between states. Nature provides the context in which these rhythms evolved. Natural light changes gradually. Dawn brightens slowly. Dusk fades. The nervous system is tuned to these transitions. Artificial light compresses them. Screens deliver intense brightness at night. Indoor spaces reduce morning exposure. The brain receives conflicting signals. It cannot clearly identify time. Nature also provides sensory patterns that regulate arousal. Wind, water, foliage, distant sounds. These inputs are non-threatening. They signal safety. They engage attention without demanding action. In such environments, stress systems quiet. Serotonin-supported regulation becomes easier. The nervous system downshifts. This is not sentiment. It is measurable. Heart rate variability increases in natural settings. Cortisol decreases. Autonomic tone shifts. Movement in nature combines multiple regulatory inputs. Light, motion, proprioception, and sensory variety align. Serotonin synthesis increases. Circadian timing strengthens. Stress hormones normalize. The effect is cumulative. It is not a single walk. It is repeated exposure. Modern life often removes these inputs. People move little. They remain indoors. They encounter light at unnatural times. They experience constant cognitive demand. The nervous system adapts. It becomes less rhythmic. Serotonin regulation loses clarity. Emotional tone becomes noisier. Restoring these inputs is not a lifestyle trend. It is biological maintenance. Morning light anchors the day. Evening darkness permits release. Regular sleep allows recalibration. Natural sensory environments reduce baseline arousal. Each of these supports serotonin's regulatory role. They do not produce happiness. They create the conditions in which stability and flexibility are possible. When these conditions are absent, serotonin's work becomes harder. The system compensates, but at a cost. Mood drifts, reactivity rises, fatigue accumulates. This is why small environmental changes often have outsized emotional effects. A consistent wake time, a brief walk in daylight, dimming lights in the evening. These are not self-help rituals. They are signals to a biological clock. They tell the system when to engage and when to rest. Serotonin listens. As this exploration continues, we will move from environment to action. We will look at how movement, touch, and human connection shape serotonin's influence, and why social and physical activity affect mood not only through meaning, but through chemistry and neural regulation. The nervous system did not evolve in isolation. It evolved in motion, in contact, and in the presence of other bodies. For most of human history, movement was unavoidable, touch was constant, and social proximity was the norm. Serotonin developed within this context. It became a chemical bridge between action, sensation, and internal state. When those elements are reduced, the system does not simply become quieter, it becomes less regulated. Movement is one of the most direct ways the body communicates with the brain. Every step activates sensory receptors in muscles, joints, and skin. These signals travel upward through the spinal cord and brainstem, informing the brain about position, effort, and balance. This continuous feedback shapes arousal and orientation. Physical activity alters serotonin in several ways. It increases the availability of tryptophan in the brain, the raw material for serotonin synthesis. It enhances the firing of serotonin neurons. Over time, it alters receptor sensitivity and network connectivity. These changes are not dramatic. They are gradual, but they accumulate. A body in motion sends a clear message. Resources are being used. Energy is flowing. The system adapts. Stress hormones rise during activity and then fall. This cycle trains the nervous system to mobilize and recover. Serotonin participates in that training. It supports the return to baseline. It helps the brain learn that activation does not have to become threat. Without regular movement, the stress system may remain chronically elevated. There is no clear period of exertion followed by release. The body stays in a state of low-grade readiness. This state feels like restlessness or heaviness. Energy has nowhere to go. Thoughts loop, muscles tighten, movement provides an outlet. It transforms diffuse arousal into structured effort. The brain receives a coherent pattern. Engage, then rest. This pattern reinforces regulation. Touch adds another layer. The skin is not just a barrier. It is a sensory organ densely packed with receptors. Some of these are tuned to pressure and texture. Others are tuned specifically to gentle, slow contact. These receptors project to regions of the brain associated with emotion and autonomic regulation. When these receptors are activated, parasympathetic activity increases. Heart rate slows. Stress hormone release decreases. The body receives a signal of safety. Serotonin interacts with this pathway. It modulates how these signals are integrated. It influences whether touch registers as calming or neutral. In mammals, grooming behavior is a primary form of social regulation. It reduces stress. It reinforces bonds. It stabilizes group dynamics. In humans, touch plays a similar role. A hand on the shoulder, a hug, sitting close. These are not symbolic gestures. They're biological inputs. They alter autonomic tone, they reduce cortisol, they shift neurochemical balance. In the absence of touch, the nervous system lacks one of its primary regulators. This is evident in infancy. Babies deprived of contact fail to thrive even when fed. Their systems cannot stabilize. In adulthood, the effects are subtler but persistent. Chronic lack of touch is associated with increased stress, sleep disruption, and mood disturbance. Serotonin's regulatory role depends on these inputs. It integrates signals of safety. When those signals are absent, the system defaults toward vigilance. Human connection extends beyond touch. The brain is exquisitely sensitive to social cues, tone of voice, facial expression, posture. These signals inform the nervous system whether it is safe to relax. Social presence reduces uncertainty. It distributes vigilance across a group. It allows individual systems to downshift. Serotonin participates in processing these cues. It influences how strongly they affect internal state. Balanced signaling allows a smile or calm voice to register as safety. In isolation, these cues are absent. The brain receives less information about external conditions. It compensates by generating its own predictions. Vigilance increases. This is not emotional loneliness alone. It is a shift in baseline arousal. Long-term isolation alters serotonin receptor expression and stress hormone interactions. The system adapts to the absence of social regulation by becoming more self-contained and more reactive. This is why loneliness feels physically uncomfortable. It is not merely sadness, it is a state of heightened internal activity without external modulation. Connection provides modulation. Conversation, even when not emotionally deep, synchronizes nervous systems. Speech rhythms align. Breathing patterns converge. Attention is shared. These processes reduce internal noise. They provide structure. Serotonin supports this synchronization by regulating how social signals influence cortical and limbic circuits. It helps translate external presence into internal stability. This is also why social environments can be overwhelming when regulation is impaired. A system already near saturation may interpret social input as threat. Noise, unpredictability, and multiple signals can overload circuits. In such states, withdrawal feels protective. It reduces input. But prolonged withdrawal removes regulatory input as well. The system loses opportunities to recalibrate. Movement, touch, and connection therefore serve as anchors. They ground the nervous system in patterns that support modulation. They do not solve problems. They alter how problems are experienced. A person who moves regularly, receives touch, and engages socially does not avoid difficulty. They process it within a system that has multiple stabilizing inputs. Serotonin integrates these inputs. It does not replace them. This perspective changes how we view interventions for emotional difficulty. It is not only about insight or belief, it is about restoring the regulatory environment in which insight and belief can operate. A mind cannot stabilize itself in a vacuum. The nervous system expects certain patterns. When they are absent, it adapts. The adaptation may be functional in the short term and costly in the long term. Reintroducing these patterns is not indulgence, it is maintenance. Even small amounts matter. A brief walk, a handshake, a shared meal. These provide signals the system understands. They tell the brain, the body is moving, the environment is safe, other systems are present. Serotonin responds. Over time, these signals accumulate. They shift baseline tone. They reduce background stress. They widen the range of accessible states. This widening does not appear as happiness. It appears as capacity. Capacity to rest. Capacity to engage. Capacity to shift. As this exploration continues, we will turn toward how modern medicine interacts with this system. We will examine how pharmacological approaches attempt to adjust serotonin's influence, what they can and cannot do, and why their effects unfold gradually rather than instantly. Modern medicine did not set out to engineer happiness, it set out to reduce suffering. The tools it developed reflect that goal. Medications that affect serotonin were not designed to create joy or personality change. They were designed to alter the operating conditions of a nervous system that had become stuck. The most widely known of these medications are selective serotonin reuptake inhibitors, commonly called SSRIs. Their basic mechanism is simple. When a serotonin neuron releases its chemical messenger into a synapse, that messenger does its work and is then taken back up into the original cell. SSRIs slow that reuptake. They allow serotonin to remain available in the synapse for a longer period of time. This does not add new serotonin to the brain. It changes how long each signal remains active. The immediate effect of this change is measurable within hours, yet the emotional effects usually take weeks. This gap puzzled early researchers. If serotonin was the mood chemical, why did mood not change right away? The answer revealed something fundamental about how the brain works. Emotional states are not produced by momentary chemical levels. They are properties of networks. Those networks adapt over time. Receptors adjust their sensitivity. Genes alter their expression. Connections strengthen or weaken. Entire circuits change how they respond to input. SSRIs initiate this adaptation. They do not impose a feeling. They alter the background conditions under which plasticity unfolds. In the first days of treatment, the brain often reacts unpredictably. Some people feel more anxious. Sleep may change. Appetite may shift. These early effects reflect the system encountering a new chemical environment. Over time, receptors regulate themselves. Some become less sensitive. Others become more responsive. Patterns of firing shift. In the hippocampus, neurogenesis increases. In the prefrontal cortex, regulatory control strengthens. In limbic circuits, reactivity decreases. These changes unfold gradually. The system learns a new equilibrium. When improvement occurs, it is often subtle. People rarely describe a sudden wave of happiness. They describe a softening. Thoughts feel less sticky. Emotions are easier to move through. A pause appears where there was once immediacy. This reflects the restoration of modulation. Medication does not replace meaning. It restores capacity. A person may still face the same life circumstances, the same work, the same relationships, the same challenges. What changes is how the nervous system holds those experiences. Problems feel more specific and less global. Discomfort feels temporary rather than defining. The future becomes representable again. This is not chemical illusion. It is change in how information is integrated. One of the most persistent misconceptions about these medications is that they create an artificial state, that they impose false contentment. In reality, they do not add experience. They alter regulation. A brain in distress often operates with reduced range. It cannot move easily between states. Medication widens that range. Some people worry that such treatment will flatten emotion, that it will remove depth. This can happen if dosage is inappropriate, or if a person is unusually sensitive. But for most, the effect is the opposite. Emotional life becomes more differentiated. Sadness remains possible. Joy becomes accessible. Reactivity decreases. The goal is not neutrality, it is flexibility. It is also important to recognize that these medications are not universal solutions. They do not work for everyone. Their effects vary. Some people respond strongly, others minimally. Some experience side effects that outweigh benefits. This variability reflects the complexity of the system. Serotonin is not one pathway, it is many. Receptor distributions differ. Genetic variations influence metabolism. Life history shapes neural organization. Medication interacts with this individual architecture. This is why treatment is often a process rather than a prescription. Doses are adjusted, different agents are tried, time is allowed for adaptation. The science behind these choices continues to evolve. Newer approaches explore specific receptor subtypes. Others target related systems such as glutamate. The field is moving away from the idea of a single chemical imbalance toward a network model. In this model, mood disorders are understood as patterns of dysregulation across multiple interacting systems. Serotonin is one lever among many. Medication adjusts that lever. It does not teach new habits. It does not resolve conflict. It does not build meaning. It creates conditions in which those processes can occur. This distinction matters because it reframes what medication can reasonably be expected to do. It is not a replacement for living. It is a support for regulation. When regulation improves, people often find that therapy becomes more effective. Reflection becomes easier. Behavior change becomes possible. New experiences integrate. Without that support, insight may remain abstract. A person may understand what would help and still be unable to act. Medication changes the cost of action. It lowers the energetic and emotional threshold required to engage. In anxiety, it reduces baseline vigilance. Thoughts become less intrusive. The body settles more easily. In depression, it reduces inertia. The future regains weight. Effort reconnects to outcome. These changes are not dramatic. They accumulate. Critics sometimes argue that this pathologizes normal emotion, that sadness and fear are part of life. This is true. But medication is not for sadness. It is for systems that cannot return. The distinction lies in flexibility. A healthy system enters distress and exits it. A dysregulated system becomes stuck. Modern mood science focuses increasingly on this concept, not on eliminating emotion, but on restoring mobility. Serotonin is central to that mobility. It influences how easily circuits disengage and re-engage. It shapes transitions. Other treatments work on the same principle through different means. Psychotherapy alters cognitive and emotional patterns. Exercise alters metabolic and stress pathways. Light therapy realigns circadian rhythm. Mindfulness alters attention and reactivity. Each acts on the same regulatory architecture from a different angle. Medication is one tool among these. It is neither a shortcut nor a failure. It is a biological intervention in a biological system. Understanding this removes some of the moral weight. It allows treatment to be seen as maintenance rather than correction. A person who uses medication is not choosing artificial happiness. They're adjusting the conditions under which their nervous system operates. The system remains theirs. The emotions remain real. The story remains authentic. What changes is the background noise. This background noise, when too loud, drowns out variation. It flattens possibility. It turns experience into a single tone. Medication lowers that noise. It does not replace music. It restores audibility. As we approach the final part of this exploration, we will step back from mechanisms and consider what all of this means at a human level. We will look at serotonin not as a molecule, but as part of the larger pattern of what it means to be a feeling organism, moving through time, adjusting to change, and carrying an inner world shaped by chemistry, experience, and care. Every moment you experience is the result of coordination. A sound reaches your ears, light reaches your eyes, signals travel through nerves, networks become active, memories join perception, predictions form, muscles adjust, breathing shifts. Somewhere inside that cascade, molecules like serotonin are released, received, recycled, and transformed. None of this announces itself. It simply happens. And from that invisible choreography, a feeling emerges. You do not feel serotonin. You feel the shape of its work. You feel it as steadiness on a quiet morning, as the ability to pause before reacting, as the sense that a difficult thought can be held without becoming a verdict, as the background impression that tomorrow exists. These are not abstractions. They are properties of a living system that is regulating itself in real time. Being human is not a purely mental experience. It is not a disembodied stream of thoughts floating in space. It is a biological process unfolding second by second. Your inner life is built on physical events, electrical impulses, chemical gradients, cellular responses. There is no separation between the mind you inhabit and the body that carries it. Serotonin sits at one of the junctions where these domains meet. It does not contain meaning. It does not know your story. It does not judge your circumstances. It participates in how your nervous system holds them. It shapes the way experience is integrated into continuity. It influences whether a moment feels isolated or part of a larger flow. This is not mechanical reduction. It is recognition. Understanding that feeling has a biological substrate does not make feeling smaller. It makes it more real. It places your inner world inside the same universe as everything else, subject to rhythm, to change, to repair. You're not imagining your moods into existence. You're living in a system that is constantly negotiating between past and present, between body and world, between threat and safety. When that system is strained, experience narrows. When it is supported, experience widens. Serotonin is one of the ways that support takes form. It is not a guarantee. It does not promise ease. It provides modulation. It provides the possibility of return from stress, return from fixation, return from collapse. Every living system depends on return. Heart rate rises and falls, breath expands and releases, wakefulness gives way to sleep and emerges again. Stability is not stillness, it is movement within range. Emotional health follows the same pattern. You do not need constant positivity, you need mobility, the ability to enter states and leave them, to feel deeply without becoming trapped, to carry memory without being defined by it, to anticipate without spiraling. Serotonin contributes to that mobility by shaping thresholds, by influencing how long signals persist, by modulating how strongly circuits engage. It does not decide what matters, it affects how mattering feels. This is why small changes in chemistry can feel like changes in identity. Not because identity is chemical, but because identity is experienced through a system that depends on chemistry to function. When the system is dysregulated, the self feels altered. When regulation improves, the self feels familiar again. Not because something artificial has been added, but because something constricting has loosened. The human brain is not a static organ. It is plastic. It changes with use. It changes with stress. It changes with care. Every night of sleep adjusts it. Every period of strain shapes it. Every moment of rest restores it. Serotonin participates in that shaping. It influences how experience becomes structure. This is why rhythm matters, light matters, movement matters, contact matters. They are not lifestyle accessories, they are regulatory inputs. A nervous system that receives regular signals of time, motion, and safety learns that it can move. It learns that activation has endpoints. It learns that the present is not permanent. A nervous system deprived of those signals adapts. It narrows, it holds, it prepares. Neither state is moral, they are responses. Care is the act of changing conditions, not forcing feeling, not commanding thought, altering the environment in which they arise. Sometimes that care is external, a routine, a medication, a conversation, a place. Sometimes it is internal, a pause, a breath, a shift of attention. These do not work by magic, they work by changing input. The system responds. You do not have to understand every mechanism, you live inside the outcome, the sense that a thought can pass, the moment when a feeling softens, the experience of being tired rather than overwhelmed. These are the system adjusting. They are not proof of perfection, they are signs of range. Serotonin does not create meaning, it creates room for meaning to shift, room for the nervous system to settle, room for the future to exist. This room is not infinite, it is maintained, it is affected by sleep and stress, by illness and loss, by connection and isolation, by rhythm and disruption. There is nothing unusual about losing it. There is nothing weak about needing help to restore it. Being human means living in a body that changes. It means inhabiting a brain that learns from everything. It means carrying chemistry that responds to experience and in turn shapes it. The quiet chemistry of being human is not a flaw. It is the medium. It allows feeling to exist at all. Without it, there would be no sadness, no relief, no interest, no memory. There would be no inner world. Understanding this does not make experience clinical. It makes it grounded. It reminds you that what you feel is happening in a living system that can move. That system is not fixed. It is not finished. It is always adjusting, even now, even here, even as this moment passes into the next one without announcing itself. As this quiet journey comes to rest, there is nothing you need to take with you. You do not have to remember names, pathways, or mechanisms. You do not have to hold on to any particular idea. All of that can drift away. What remains is simpler than information. The sense that your inner world is not random, that what you feel has structure, that your mind and body are not separate forces pulling in different directions, but one living system learning how to stay in balance. Somewhere inside you, tiny signals are still moving. Cells are still exchanging messages. Patterns are still shifting. Even in stillness, change continues. You do not need to manage it. You do not need to supervise it. Much of this work happens without your awareness, the same way breathing continues when you stop thinking about it. If your day has been heavy, it is allowed to loosen here. If your thoughts have been loud, they can soften. If your body has been holding tension, it does not need to hold it all at once. Nothing has to resolve tonight. There is no conclusion required, just a gentle return to the rhythm that carries you forward without effort. Thank you for spending this time here. Thank you for letting curiosity slow down instead of speeding up. Thank you for being part of this quiet space. You can let the sound fade. You can let your attention drift. You can let rest arrive in whatever way it does, and the system inside you will keep doing what it has always done, adjusting, balancing, continuing.